So I want to talk about the flora. What the California flora is, an incredible gift to the universe. It is so diverse. And part of its diversity is this interesting coastline we have. Without being a jerk or anything, Oregon has a very straight coastline and they don't have as many seaweeds as we do. So just the diversity of habitats in all these bays and wrinkles here, and these prominent bumps that cause currents to move around them in an interesting way, increases diversity here. So some of my favorite places to collect are Bodega Bay, where I first learned, Point Arena, north of that in Northern California. San Francisco Bay is rich in seaweeds, and there are some puzzlers in there. Monterey Bay, San Luis Obispo. For a long time, there was no highway there. And it was only until after World War II that people could actually get to the coast easily and collect. One of the big breaking points that changed this rather north-south facing coastline is Point Conception. Very, very rough oceanic place. I have not collected there, and that's one of my goals in life. But from Point Conception, the coastline changes and goes east into the Southern California Bight and the 8 islands within that bight. Those are some of the places that I've loved the most to collect. I've spent many, many years and many, many hours underwater in the Channel Islands, the northern, mid, and the southern ones. Places where seaweeds grow in abundance because they haven't been as impacted by human activities. Thank goodness there's lots of marine protected areas along our coast. It was a big battle in the last few decades to protect coastline from humans, from this incredibly opportunistic and successful species that we are. So you have to be careful when you go to a place where collecting is available. San Francisco Bay is also, there's places, even if you just go to the marinas and look at the floating docks, you can see a lot of seaweed that way. An incredible amount of non-native seaweeds as well. If you go a little bit further north to the Sonoma coast, and it's about an hour north, places north of Bodega Bay, which is a protected area, like Schoolhouse Beach and other little pocket beaches, Goat Rock, those are open. To my knowledge, you should check before you go, but they're beautiful. They're beautiful pocket beaches with good seaweed flora. So here is a temperature map of our coast, from cold temperatures in the reds to purples to blues, to warmer temperatures in the greens, and then higher temperatures in the orange. So you can see as we come down into British Columbia and Alaska, we're all in the very cold waters, both offshore and near shore. We get this continues down into Northern California, which we kind of define by north of San Francisco Bay. We get lots of cold water. And as we go south, here's Point Conception, this mid-Pacific warmer water reaches our coastline. So California is really half or 1/3 cold water influence. The central is sort of mixed. It's sort of a gradient of cooler getting to warmer. And then we have the warm Southern California Bight. Here's a closer up view of that. Here's Point Conception, here's San Francisco Bay and Point Conception. So you can again see this cold water here and how very much warmer it gets as you get into the Bight. But look at these little pockets of cold here, of the blue colors. That is upwelling. The upwelling of cold bottom water, nutrient-rich bottom water. When the winds blow offshore, this water is brought up to replace the surface water and brings cold water and its ability to hold nutrients and all the nutrients that were derived from the compost heap at the bottom of the ocean. So it's really these upwelling events down here in Southern California and all the way down Baja California Sur that allows the kelps, which require cold water and a lot of nutrients to make their bodies. It allows them to grow far further south than the cold water zones up north. Let's look at this a little bit more closely. Here's another view of temperatures, again, from cold to warm. You can see that the 8 Channel Islands lie in very different temperature regimes. The northern islands, as you go east, are warmer. As you go west, the cold water from the north affects them. These islands are in warm water. They're very similar to Mexican waters. This is the farthest west one of the lower islands, and it gets a little bit of cooler water too. So each island is in a different temperature regime. These currents are so important to establishing these temperatures. Here's the cold California current coming down. Here are the northern islands. There's a warm counter current coming up from the tropics, and there is warm water from the Central Pacific. And it all gets mixed in the northern part of the Southern California Bight. So it's a very dynamic place because the temperatures are so variable, and temperature has a lot to say about where species can exist. Upwelling on this map too, very, very important. So the cold temperate biogeographic province is that which is north of San Francisco Bay. And that's where you get your kelps. And if you look underwater, the seaweeds there are large. These are reds. They are dark and pigmented because those red pigments require nitrogen. They're made of nitrogen, and cold water holds nitrogen. These are make a beautiful deep understory of blades, crusts. Oops, I'm pointing with my finger. Crusts, blades. Look how deep and dark they are. Calcified reds. So here's just a bunch of views of these crustose corallines. I'll talk about a little bit more later. Beautiful subtidal seaweeds that are typical of cold water. But now let's look at below Point Conception, this part of California. Looks a lot different. Warm water doesn't hold the nutrients. Things are paler. They are not as large. They're more densely, roughly, turfy. So this is just a view in the edge of a macrocystis forest here. Typical subtidal view in Southern California. How different than the slide I just showed you. Some species that grow down here in the understory, again, they're shorter, paler, and really, really intricate. These turfs can hold hundreds of species, many of them poorly known. You can just tell intuitively how different that warm water habitat and biogeographic province is. And much of it is derived from temperature or maintained by temperature and history. So the California seaweed flora, the book, The Marine Algae of California by Abbott and Hollenberg, was published in 1976, the year I took my first course. And in that book was 669 species. And look what's happened now to date. 500 names have changed, which is, to me, astounding. And it's because we've learned more about those species and their relationships to each other. We've moved species into new genera because they're actually not related to the old ones. There's many different reasons why the names have changed, but this activity has been promoted by more knowledge and especially using DNA sequences as characters. 130 taxa have been added, and those taxa are ones that have been discovered because of technology. For example, the ability to go deeper. Seaweeds only can go as deep as light penetrates, but light penetrates quite a bit in some places, especially Central California and Southern California. So seaweeds, the deep water seaweeds have been added to the flora, and species we now know that are not native to California have been introduced to California and have added to our flora. So where were these species described? Try to move this thing, let's see if I can. 361 species have type localities, that is the original type specimen on which the species is based, was collected in California. And that is partly a legacy of Setchell and Gardner, who just cleaned up with all their helpers who were collecting everywhere. They named a lot of seaweeds. A lot of their names have changed too, but still a lot of Setchell and Gardner seaweeds remain. And of course, many, many other great phycologists have collected in California and named species too. 60 have the type specimen was collected in Mexico. And that's not surprising because there's just an artificial boundary between Mexico and California. We're really one continuous warm water current influenced place. So we do share a lot of species with Baja California, Mexico, Pacific side, a few from the Gulf as well. 35 seaweed species have type localities in Asia, Japan especially, because the Japanese were far ahead of us in describing species for Japan, and they named a lot of species. Sometimes we thought our species matched theirs, therefore we called them Japanese species. And we're finding out some of them aren't Japanese species, they're California species. Still, as I show you, we do share species with Japan. And this is the kicker, I think. 250 species in our flora have type localities in other parts of the world, especially Europe, other oceans.
Kathy, you're muted again.
Good, because then you didn't hear me say, how do I change the slide? Now I've
Okay.
changed it. Okay, thank you. Please tip me off as you are. So let me examine. So I told you where the type localities are because that's where the humans come in. The only way we know these things is because we're humans who study them. So we're very much a part of the California flora because we're the ones who are studying it, creating it. There are some species on both sides of the Pacific, Japan, Korea, and California, that are the same. And this is probably because the shape of the Pacific Ocean has changed tremendously over the last few million years. And it could be that the currents, the warm Kuroshio current, the colder northern currents coming down the coast have moved species across the Pacific. It's also possible that tsunamis have moved species across the Pacific, mostly from Japan to us, just as we saw with tsunamis in our time. A lot of wrecks came over here. Evidently, throughout Japanese history, their wooden ships, when wrecked, could float all the way to California, bringing with them a load of species. So we do share some species on both sides of the Pacific. Further, in the tropical zone has been a much more stable zone on the Earth around the equator. The Great Tethys Sea was a band of ocean that was pretty much connected at one time in the history of Pangaea, etc. And so warm, temperate, and tropical species have moved around the globe. So here's an example. Cytosiphon lomentaria, Dactylosyphon bullosus, Dictyota coriacea, Dictyo neurum undulatum, Terracladiella, Capillacea. All of these occur in Japan. Chetomorpha spiralis. These are all species that occur on both sides of the Pacific. Only a few of them have been studied to see which side they originated on or their phylogeographic history. Where did they come from? How did they travel? So there's still a lot to be learned about the connection between the Western and Eastern Pacific. I am unmuted. Species, of course, evolved here, and they are native species. Here's one that I studied that was interesting to me because the project originated in Japan. They wrote to me and asked me for specimens of Chondria tenuissima, a Japanese species. And I said, we have a Chondria here. Can I send you some of ours? And they said yes, and we studied together. It turns out that Chondria, which is a genus that was described in Europe, it's from England, is not in Japan. And so we named the Japanese and the California species Neochondria, a new chondria that's completely unrelated to the chondrias of Europe. So this is Chondria nidifica here, now Neochondria nidifica. It is a native to California. It's found nowhere else. Originally described from San Diego. But there are other chondrias in California too, and they need to be studied. Here's one, Chondria acrorhizophora. Anyone from Southern California who's looked in tide pools and seen this purple? It's a gorgeous seaweed. Needs to be revisited. And there are a couple other chondria species on this coast to see if indeed chondria actually moved around the world evolutionarily, or if these are unique species, unique to California. As I said, the ocean is not a static environment. Over evolutionary time, the continents have moved, the currents have changed, and seaweeds have moved too. So some of the seaweed ranges have expanded over time just in a stepwise fashion. Gloriopeltis furcata is a species that has its greatest abundance, this genus, in Asia, Asian Russia, Korea, and Japan. So it is very common in Northern California where it gets large in the intertidal. But in Southern California, there's a great disjunction. Central California does not have this species, a disjunction, and then it shows up as this tiny little turfy thing. Is it just temperature? And I said, well, maybe they've diverged. Maybe they're not the same species. But you know what? I worked with people from Korea and Russia and Japan. It's all one species, all the way down from Kamchatka, all the way down our coast to southernmost California, Catalina Island. Unbelievable to me, but a story the seaweeds can tell about how they occupy their native range. Native ranges change through evolutionary time. Species ranges are expanding in our own time, and I wanted to tell you just one story of one that I've seen in my own long life. This is Asparagopsis taxiformis, the cotton candy weed. Maybe you've heard about it because this is the one that people are studying and trying to raise to feed to cattle to dampen methane production. You heard that study? This is the species. It's all over the tropics. It's in Hawaii and it's in southern Mexico. When I first started looking underwater in the Southern Channel Islands. This was in the early 1980s. When I saw this, it was extremely exciting. And I knew from the literature that it had two phases, a haploid phase that was cotton candy and a diploid phase that's like little puffballs, tiny filamentous puffballs. So I saw a few of these and had to search and search and search for the alternate phase. I'll be talking more about phases, don't worry. Now, this is everywhere down there. Its range has extended tremendously in Southern California, especially in the islands where there's less human intervention. So this is a story of warm water species moving north with warm water. Probably every El Nino gives it a boost and a chance, and either it can make it or it can't. It'll try again. This is an odd one. It's a species of Padina, which is a tropical species that a colleague of mine who's doing intertidal monitoring found in Marina del Rey in Southern California. The only time it's ever been seen. And I just contacted him and said, would you look for that thing again? I want to know if it's still around. So it has nothing like that has been seen in California, but it's probably around. Ranges expand in our own time. Some species we thought were native, but in fact have later been proven to be non-native, but they're here and they've been here for ages. Sargassum muticum came to this coast on oyster spat in about 1950. It was in Southern California by 1970. It just raced down the coast. And it's everywhere, even in a few places on the open coast. Many of the non-native species are in harbors or in human kind of places where the ships come in and can deliver spores, deliver fragments, deliver whatever. So Lomonteria hackadensis, clearly from Japan, was in Marine Algae of California as a native because it's been here. We don't know when it came. Possibly it too came with oysters, but we don't know its origin story. Gracilaria vermiculophila is not in Marine Algae of California, but there are a couple species of Gracilaria that have been mistaken for this species. This is a non-native Asian species that is in fact in almost every harbor in California. And it was identified as a native, misidentified. So there are many species that have come here in the past before we've been able to observe them, and they've made themselves at home and become part of the flora. Worked with the herbivores, worked with where they could survive, and they're part of our flora. Some conspicuous introductions, Sargassum horneri. These were introductions that came in my lifetime. In fact, Sargassum horneri was found as a very small plant at Long Beach Harbor. I had a Japanese phycologist visiting and he recognized it. Now it is all over Southern California and considered quite a pest because it's wiry, it gets caught in anchors, it is taking up a lot of room, it's competing with other natives. Undaria pinnatifida. I was working at Catalina Island when a halibut fisherman came in with a seaweed and said, "What's this?" And I said, "I do not know. Where did you get that thing?" And it turned out to be the Japanese kelp that we eat in miso soup, very common in Japan, raised in Japan. It's even thought that the cultivation in Japan and Korea of Undaria pinnatifida has created some races that are very good at moving around because they've selected them for eating. So Undaria pinnatifida is on our coast. It hasn't raced around as much as Sargassum horneri, but it's in San Francisco Bay, for example. You can find it on the docks. There's been some removal programs, and it's part of our flora now. More recently recognized introductions. These are both that are not in Marine Algae of California, 1976. And they've always fascinated me because they grow in these sandy environments, muddy, sandy environments where a lot of native seaweeds don't grow. I was working on another project looking at Zoster marina. So I spent a lot of time in these soft sediment substrates and kept seeing these two seaweeds. Studied this one and found that it has recently come from Japan. There's very little genetic difference between Japanese populations and California populations. This one I've always predicted is non-native, and we're just now finishing work demonstrating that Tynocladia crassa is a non-native seaweed that has established, like Mutamo, in these soft sediment places with less competition from the natives. And then finally, species we once thought were in other parts of the world, California species, are not. Paikia californica was reported from Japan and Korea, but in fact, it's another species, Yoshizaki, that just looks a lot like Paikia californica. The morphologies are very similar, but they're not genetically close. They're in the same genus, but they're not the same species. Strangely, Paikia californica has gotten to Great Britain. Some people think on the pontoons of float planes or something like that during World War II. Who knows? It's hard to track that down. Similarly, the genus Rhodymenia, flat dichotomous blades. Rhodymenia californica has been reported all over the world. This is Rhodymenia californica, as reported in Peru. But in fact, all of these are one species of Rhodymenia, Rhodymenia corallina. Rhodymenia californica is not in Peru. So this is how we can tease apart origins of seaweeds using DNA sequence evidence. So going back here, these are where seaweeds were described, but we don't really know what's native. And I find that intriguing and wonderful. And there's generations of study to be done to determine who our seaweeds are related to and what kinds of mistakes we've made in the past, mostly due to morphological variation and morphology failing us as a way of telling these species apart. DNA sequences are very important. So our seaweed flora is a mosaic of native and naturalized and recently, that is in our own lifetimes, introduced species. And seaweeds that are attached, that are sessile, actually move over various timescales. And it's likely that climate change and current change and ocean change is a driver of moving species around the oceans. Phylogeography, using DNA sequences to match populations in different places to find the native versus the introduced population, to determine origins and dispersal, very, very important frontier. But as I say, there are lots of puzzles still, and every species has a story. You cannot generalize, which is frustrating and delightful.